Welcome friends to new session on molecular biology. First, we will discuss translation in eukaryotes and post-translational processing. We have already discussed the process of protein synthesis in prokaryotes. Mechanism of protein synthesis in eukaryotes is similar to that of prokaryotes. The differences are due to the more complex cellular organization of the eukaryotic cell. The protein synthesizing message is present in mRNA. Ribosomes read these messages into protein. The connection between message to protein is made by some adaptive molecules called amino-acylated tRNAs. They are also called charged tRNA. Amino-acyl tRNAs are tRNAs to which a specific amino acid has been attached. This linkage is catalyzed by an enzyme called amino-acyl tRNA synthetase using ATP molecules. In eukaryotes, protein synthesis occurs in 4 different stages: activation of amino acids, initiation, elongation and termination. However, there are some significant differences, particularly during initiation. In eukaryotes, each mRNA is monocistronic, that is, the mRNA encodes a single protein. In prokaryotes, many mRNAs are polycistronic. That means they encode several proteins. Each coding sequence in a prokaryotic mRNA has its own initiation and termination codons. Let's discuss on the various steps in translation. First, activation of amino acids. For protein synthesis, amino acids are selected from the amino acid pool of the cytoplasm. Before incorporated into protein, amino acids get activated by ATP with the help of enzyme amino-acyl tRNA synthetase. This enzyme is highly specific in identification of both amino acids and tRNAs. There is at least one tRNA for each of the 20 amino acids. After ATP mediated activation of amino acids, the carboxyl group of amino acids is attached to 3' hydroxyl group of tRNA with a ester linkage. Please see the figure on screen, which explains how the amino acids is attached to 3' hydroxyl group of tRNA with a ester linkage. Initiation. This is the first step in translation. Please see the figure on screen, and this involves 3 steps. First one, recognition step. Second one, formation of 43S pre-initiation complex, and third one, formation of 80S initiation complex. In recognition step, the first AUG triplet after the marker nucleotide is identified by the ribosome as initiation codon. In mammals, the marker sequences are called as Cossack sequences: 5' A, C, C, A, U, G, G, 3'. Initiation factors are required for this process. The 43S pre-initiation complex consists of the following molecules: A strand of mRNA, B initiation factors, C 40S ribosomal subunit, D methionine tRNA, and E energy in the form of GTP and ATP. Let's discuss these molecules one by one. First one, mRNA. Eukaryotic mRNAs are monocistronic and use only one initiator codon, that is AUG. These RNA molecules consist of Cossack consensus. Then initiation factors. Eukaryotes use many initiation factors for starting the process of translation. The important eukaryotic initiation factors are EIF3, EIF4A, EIF4B, EIF4C and EIF4F, in which E means elongation and IF means initiation factors. 40S ribosomal subunit. It is the smaller subunit of eukaryotic ribosome. The 40S ribosomal subunit is linked by EIF4C, that is elongation initiation factor 4C, which may be helpful for the association of 2 ribosomal subunit. Now, methionine tRNA. The initiated tRNA carries an unmodified methionine to the 40S initiation complex. Eukaryotes contain two structurally different tRNAs that recognize a UG codon that means one for methionine for the initiation and the other for methionine for the inner part of protein. The initiator codon tRNA is recognized by tRNA I and non-initiated AUG codons are recognized by AUG M. Same methionine tRNA synthetase is responsible for adding methionine residues to tRNA I and tRNA M. The initiated tRNA in eukaryotes carries an unmodified methionine, that is not formylated as in prokaryotes. The initiation factors, that is EIF4A, is a complex molecule that consists of a subunit that acts as a cap binding protein, that is CBP. The CBP may initiate mRNA binding on ribosome by interacting the 5' cap region present on mRNA. CBP promotes binding of EIF4A, EIF4B to mRNA. These initiation factors and ATP are helpful for unbinding of secondary structure in the 5' untranslated region of mRNA and formation of a 43S initiation complex. After attached to the 5' end of the mRNA, the initiation complex identifies initiation codon, that is 5' AUG 3' based on the presence of Cossack consensus sequence. Now, formation of the 80S initiation complex. The joining of the 60S subunit to the 43S initiation complex to form 80S initiation complex requires the action of EIF5 and EIF4C. All other initiation factors are removed upon completion of formation of the 80S initiation complex. Elongation of translation in eukaryotes. Ribosomes are the sites of protein synthesis. The whole ribosome consists of three sites, P, A and E sites respectively. P site or peptidyl site carries the peptidyl tRNA. The site carries growing peptide chain. A site or amino acyl site carries new incoming tRNA with the amino acid to be added to the growing peptide chain. The E or exit site is occupied by deacylated tRNA. Elongation process occurs by a similar mechanism in eukaryotes as in prokaryotes. Elongation has three steps. First one, binding of amino acyl tRNA to A site. This step involves attachment of a new amino acyl tRNA to A site. The next codon in mRNA determines the second amino acid. The elongation factor EF1 and GTP are involved in this process. Second one, formation of peptide bond. In this step, the amino acid present in the A site of ribosome forms a peptide bond with the amino acid in the P site. This reaction is catalyzed by the enzyme peptidyl transferase. This enzyme is 28S rRNA, a component of 60S subunit. Third one, translocation of ribosomes on the mRNA. After the peptide bond formation, the uncharged initiation tRNA leaves the P site and the peptidyl tRNA moves to the new vacant P site with the two amino acids attached. The ribosome advances 3 bases to read the next codon and the process repeats. Energy in the form of GTP is utilized for the translocation process. Termination of translation. It is the last step in protein synthesis. Termination occurs by a similar mechanism in eukaryotes as in prokaryotes. Termination continues until a termination codon is encountered. Transfer RNAs do not pair with stop codons, that is UAA, UAG and UGA. The stop codons are identified by release factor. When the release factors bind to ribosome, translation termination occur. Termination is effected by peptidyl transferase activity of the ribosome. GTP hydrolysis is required for termination and dissociation of ribosomal subunit. The dissociated ribosome subunits enter the cytoplasm where they remain until used again in another round of translation. Regulation of RNA stability. Alterations in RNA stability may take a considerable contribution to gene regulation in eukaryotes. Regulation of RNA stability may operate in either nucleus or the cytoplasm. Now, regulation at nucleus. Some genes are known to produce heterogeneous nuclear mRNA, that is HNmRNA, which in some cells is never processed to mRNA and never transported to the cytoplasm. These transcripts are degraded in the nucleus before they can be processed. Now, regulation at cytoplasm. The stability of mRNA can control the availability of the mRNA for translation in cytoplasm. The stability of mRNA of different genes may vary considerably in their half-life. Some genes that codes for abundant product with a long duration of action, such as the beta hemoglobin gene, may produce mRNA that has a long half-life. Other genes that codes for a product with a short duration of action, such as those of growth factors, may produce mRNA that has a short half-life. Many growth factor mRNA have half-lives of less than 1 hour. Sequences within the 3' untranslated region of unstable mRNA that are responsible for their rapid degradation have been identified. There is also strong correlation between length of poly A tail and the half-life of the mRNA. The longer the poly A tail, the longer the half-life of the mRNA. Now, let us see some examples of gene regulation by RNA instability. The stability of alpha and beta tubulin mRNA may change depending on the availability of alpha-beta dimers. The more alpha-beta dimers are available, the less stable are alpha and beta tubulin mRNA. If tubulin polymerization is inhibited with colchicines, the amount of alpha-beta dimers increases. Further, alpha-beta tubulin dimer production is stopped through selective destabilization of alpha and beta tubulin mRNAs. Now, inhibitors of protein synthesis. Different types of compounds inhibit protein synthesis, such as therapeutic drugs, toxins and plant products. Let us discuss these molecules one by one. First one, therapeutic drugs. A number of antibiotics that inhibit protein synthesis have proven to be effective antibacterial drugs. Some examples are A. Streptomycin. It binds to 30th subunit of bacterial ribosomes. It causes misreading of mRNA and at high concentration. They completely inhibit initiation complex formation. B. Tetracycline. It binds to the 30th ribosomal subunit and inhibits the binding of amino-acyl tRNAs. C. Chloramphenicol. It binds to the 50th ribosomal subunit and blocks the peptidyl transferase reaction. D. Erythromycin. It is an antibacterial drug that inhibits the translocation reaction by binding to the 50S ribosomal subunit. E. Rifamycin. It blocks initiation transcription by binding to RNA polymerase, which prevents RNA synthesis. Second, inhibitors. Other antibiotics inhibit protein synthesis and are used for research purpose but have not proven to be good therapeutic drug. These include A. Cyclohexamide. It inhibits protein synthesis in eukaryotes by binding to the 60S ribosomal subunit and inhibiting the peptidyl transferase reaction. B. Puromycin. It is an analog of the amino-acyl adenosine moiety of amino-acyl tRNA and leads to premature termination of translation in both prokaryotes and eukaryotes. It competes with amino-acyl tRNA for binding to the A site. In the A site, peptidyl transferase links its amino group to the carboxy group of the peptidyl tRNA in the P site. This linkage causes premature termination. Third one, toxins. The mechanism of action of several potent toxins is through the inhibition of protein synthesis. A. Diphtheria toxin. It is produced by a lysogenic bacteriophage that infects Corynebacterium diphtheriae. Diphtheria toxin causes inactivation of EF2 by attachment of ADP to EF2 and inhibits translocation step of elongation. Because diphtheria toxin is catalytic, only small amounts are needed to be toxic to the cell. B. Ricin. It is a protein produced by castor beans. The ricin toxin is N-glycosidase and removes a single adenine from 28S rRNA. This depurination inhibits protein synthesis in eukaryotes. C. Ribosomal RNA specific nucleases. Other toxins inhibit protein synthesis by the specific cleavage of rRNA. Alpha-sarcine is a toxin produced by the fungus Aspergillus giganteus, which cleaves 28S rRNA. This specific cleavage leads to the inhibition of amino-acyl tRNA binding. Colocin E3 is another example of ribosomal RNA specific nuclease. It is secreted by some strains of E. coli, inhibits protein synthesis in other bacteria. It does this by specifically cleaving 16S rRNA, which leads to the inhibition of initiation. Now, post-translational processing of proteins. Translation is not the end of the genome expression pathway. The polypeptide that emerges from this ribosome is inactive and before taking on its functional role in the cell must undergo post-translational processing. In eukaryotic cell, most protein synthesis occurs in cytoplasm. The remainder of the protein synthesis occurs on the rough endoplasmic reticulum. Proteins synthesized on RER, that is rough endoplasmic reticulum, require transport across a membrane before they reach their final destination. It has been discovered that the ultimate cellular location of proteins is often determined by specific, relatively short amino acid sequences within the proteins themselves. These sequences can be responsible for proteins being secreted, imported into nucleus or targeted to other organelles. Let's discuss on protein targeting. Proteins to be synthesized on endoplasmic reticulum have a characteristic leader or signal sequence that directs their synthesis to ER. This process is called as protein targeting. Signal sequence present at amino terminal end of protein. It is usually about 20 amino acid length. Different steps involved in protein targeting are: first one, recognition of signal sequence by signal recognition particle, that is SRP. Second one, SRP ribosome directed protein synthesis into the ER. Third one, completion of synthesis into the ER. Now, let us discuss each step one by one. First one, recognition of signal sequence by signal recognition particle, that is SRP. The proteins synthesized on ER are made in a precursor form. During translation, protein emerges from the ribosome. It is identified and bound by a SRP. SRP is a nucleoprotein consisting of 6 polypeptides and a 7S small cytoplasmic RNA. Second one, SRP ribosome directed protein synthesis into the ER. SRP ribosome complex arrests further elongation of polypeptide chain by blocking ribosome. The SRP ribosome complex interacts with a SRP receptor complex in ER. The interaction of these two complexes, that is SRP ribosome complex and SRP receptor complex, insert signal sequence into the membrane and restarting of protein synthesis. Just prior to restarting of protein synthesis, SRP is cleaved from ribosome using ATP. During insertion of signal sequence into the membrane, the signal sequence binds with another membrane protein called signal sequence receptor, SSR. Third one, completion of synthesis into the ER. While signal sequence is attached on SSR and remainder of the protein is being synthesized, a signal peptidase on the luminal side of ER cleaves the signal sequence from protein. The protein passes through the membrane into the lumen during synthesis. A number of changes in newly synthesized proteins occur in the ER and Golgi complex. The major types of post-translation processing are: first one, protein folding; second one, proteolytic cleavage; third one chemical modification, and fourth one Intein splicing. Often these different types of processing occur together, the polypeptide being cut and modified at the same time that is folded. If this is the case, then the cutting, modification or splicing events may be necessary for the polypeptide to take up its correct three-dimensional conformation, because this is dependent on the relative positioning of the various chemical groups along the molecule. Let us discuss about these concepts. First one, protein folding. When a protein is being synthesized, it may assume a three-dimensional structure for attaining biological activity. The three-dimensional folding up of newly formed proteins are regulated by a family of proteins collectively called chaperones. Please see the figure on screen. Chaperones helps to produce the correct spatial arrangement of a nascent protein. They attach a newly formed protein and prevent wrong folding, so that folding is directed only in the correct conformation. Chaperones are diverse family of proteins. Some of them are sensitive to heat, hence called heat shock proteins or HSPs. Production of HSPs increase when the cell exposed to various stress inducing responses such as radiation, free radicals, heavy metals, bacteria, etc. Hence, they are also called as stress proteins. HSPs are classified into various families based on their molecular weight. The main HSP family proteins are HSP10, HSP60, HSP70 and HSP90. In these families, HSP60 is also called chaperonin, is well studied chaperone. In this figure, we can see the action of chaperones during translation. Next, proteolytic cleavage. It has two functions in post-translational processing of proteins. First one, it is used to remove short pieces from the N and or C-terminal regions of polypeptides, leaving a single shortened molecule that folds into the active protein. Second, it is used to cut polyproteins into segments, all or some of which are active proteins. A similar type of processing occurs with insulin, the protein made in the islets of Langerhans in the vertebrate pancreas and responsible for controlling blood sugar levels. Insulin is synthesized as pre-proinsulin, which is 105 amino acids in length. The processing pathway involves the removal of the first 24 amino acids to give proinsulin, followed by two additional cuts which excise a central segment, leaving two active parts of the protein, the A and B chains, which link together by formation of two disulfide bonds to form mature insulin. Please see the figure on screen, which shows the proteolytic processing of insulin. The first segment to be removed, the 24 amino acids from the N terminus, is a signal peptide, a highly hydrophobic stretch of amino acids that attaches the precursor protein to a membrane prior to transport across that membrane and out of the cell. Signal peptides are commonly found on proteins that bind to or cross membranes in both eukaryotes and prokaryotes. Third, chemical modification. Chemical modification is another important post-translational modification involved in eukaryotes. This type of modification involves incorporation of small chemical groups such as acetyl, methyl or phosphate to the amino acid in a protein. Chemical modifications enable the protein molecules to perform specific biochemical function. Most important types of chemical modifications are glycosylation and addition of lipids. Let us discuss the details of these concepts. A. Glycosylation. It is a complex type of modification in which large carbohydrate side chains are attached to polypeptides. There are two general types of glycosylation. O-linked glycosylation. It is the attachment of a sugar side chain via the hydroxyl group of a serine or threonine amino acid. Please see the figure on screen. N-linked glycosylation. It involves attachment through the amino group on the side chain of asparagines. B. Attachment of lipids. In eukaryotes, the protein molecules targeted to the plasma membrane are modified by the attachment of lipids to the growing polypeptide chain. Generally, there are three types of lipid additions: A. N-myristylation, B. Prenylation, C. Palmitylation. Now, N-myristylation. The process of attachment of fatty acids to the N-terminus of the growing polypeptide during translation is called N-myristylation. Now, prenylation. It is a process of attachment of lipids on nascent protein molecules. In prenylation, a specific class of lipids called phenyl groups are attached to the sulfur atoms in the cytosine residues of a polypeptide chain. Many plasma membrane associated proteins involved in the control of cell growth and differentiation are modified in this way. Now, palmitylation. In the third type of fatty acid modification, palmitylation, palmitic acid, a 16-carbon fatty acid, is added to sulfur atoms of the side chains of internal cysteine residues. Now, Intein splicing. This is another type of post-translation processing. This process is almost similar to intron splicing seen in pre-mRNA. Inteins are internal segments of proteins. During Intein splicing, the internal segment of proteins are removed soon after translation, then the two external segments or exteins becoming linked together. Now, let us summarize the session. Translation in eukaryotes is divided into three distinct activities: initiation, elongation and termination. Initiation includes the precise attachment of a small ribosomal subunit to the initiation codon of mRNA, which sets the reading frame for the entire translation process, that is, the entry into the ribosome of a special initiated tRNA and the assembly of the translational machinery. During elongation, a cycle of tRNA entry, peptide bond formation and tRNA exit repeats itself with every amino acid incorporated. Amino acid tRNAs enters a site where they bind to complementary codon of the mRNA. After tRNA enters, the nascent polypeptide attached to the tRNA of the P site is transferred to the amino acid on the tRNA of A site, forming a peptide bond. Peptide bond formation is catalyzed by a portion of large ribosomal RNA acting as a ribosine. In the last step of elongation, the ribosome translocates to the next codon of mRNA, as the deacylated tRNA of the P site is transferred to the E site, and the deacylated tRNA that was in the E site is released from the E site. Translation is terminated when ribosome reaches one of the three stop codons. After ribosome assembles at the initiation codon and moves a short distance towards 3' end of mRNA, another ribosome generally attaches to the initiation codon so that each mRNA is translated simultaneously by a number of ribosomes. The complex of an mRNA and its associated ribosomes is a polyribosome. The secretory proteins are synthesized in rough endoplasmic reticulum. The newly formed proteins are directed to endoplasmic reticulum by a signal peptide. Carbohydrate moieties are added at particular region of newly formed protein for its delivery to targeted sites. Proteins may be post-translationally modified in a variety of ways. Proteolytic cleavage, usually by specific peptidases, activate proproteins. The signal peptides of the proproteins are removed by signal peptidases. Covalent modifications alter many types of side chains in a variety of ways that modulate the catalytic activities of enzymes, provide recognition markers and stabilize protein structures. Protein splicing occurs via the Intein catalyzed self-excision between an N-extine and C-extine via peptide bond. Now, you can work out the assignments given here. First one, describe eukaryotic translation. Second one, discuss translation inhibitors. Third one, describe post-translational modifications in eukaryotes. Fourth one, describe signal hypothesis. Fifth one, explain the role of molecular chaperones. Now, I can suggest some books for your further studies. First one, Biochemistry, written by Berg J.M., Tomosco J.L., Stryer L., 5th edition, published by W. H. Freeman and Company, New York, USA, 2008. Next, Cell Biology, Karp G., 7th edition, published by John Wiley and Sons, Singapore, 2014. Next, Molecular Biology of the Cell, written by Alberts B. Johnson A., Louis J., Ralph M., Roberts K., Walter P. 5th edition, published by Garland Science, New York and London, year 2007. Gene 9, written by Levin B., published by Johns and Bartlett Publishers, Sudbury, Massachusetts, 2007. Next, Biochemistry, written by Davidson B.L. and Sitman D.B., year 1997, published by Waverly, New Delhi. Next, The Mechanism of Eukaryotic Translation Initiation and Principles of its Regulation, written by Jackson R.J., Helen Cutt and Pestova T.V., year 2010, published in Nature Reviews Molecular Cell Biology, 11th edition, page 113 to 127. These are the few websites which you can refer. First one, www.higher.mcgraw-hill.com. Second, www.themedicalbiochemistrypage.org/proteinsynthesis.php. Thank you for watching this program. Let's meet in another session with a new topic. Bye.